Hey biology students! In this video, we're going to cover molecular genetics in one of a few different videos. In this video, we're going to cover DNA structure. First, let's talk about the historical discovery of the structure of DNA involving these scientists. The story goes that Rosalind Franklin and Maurice Wilkins worked together in a lab at King's College in London in the 1950s. Now, imagine this, being a woman in science in the 1950s working in a lab was a very unusual thing. And she did really excellent work. However, she and Maurice didn't really get along, but they did happen to have the best data as far as what the structure of DNA looked like. They worked with a type of lab technique called X-ray crystallography, which means being able to take photos of very, very small molecules, and DNA is a very small molecule. This is their picture that they generated in their lab, and it shows the DNA is a double helix. There's two strands of the DNA molecule. So James Watson and Francis Crick, they went to a secret meeting with Maurice Wilkins where they saw this data that Rosalind had generated, and they used that information to come up with the structure of DNA as double helix. Then in 1962, the Nobel Prize was awarded. And the Nobel Prize is the most prestigious award you can win in science. It comes with lifelong fame as well as $1 million cash. So the Nobel Prize for the structure, determining the structure of DNA as a double helix, this prize goes to James Watson, Francis Crick, and Maurice Wilkins. Notice Rosalind Franklin is left out. Well, she actually died. She died in 1958, actually of cancer, likely because she worked with X-rays in the lab. And back then, of course, they didn't realize that X-rays can cause cancer. That's a type of radiation that can damage your DNA and cause cancer. And she had been exposed to large quantities of X-ray radiation as part of her lab research. So the Nobel Prize can only be awarded to people who are alive. So she was not given recognition in the Nobel Prize. And she was actually left out of biology textbooks for almost about 2 to 3 decades. It wasn't really until the 1980s that her picture started showing up in biology textbooks, giving her the recognition she deserves for her work that also probably killed her. So thumbs up to you, Rosalind Franklin. So the DNA structure, if we take a look at the chemical molecule, DNA is an example of a polymer. If you recall, we used this terminology at the beginning of the semester to describe large macromolecules in biology as polymers, and those polymers are made out of smaller subunits or monomers. And in this case, DNA is a polynucleotide. So we use that word polynucleotide, poly meaning many. So DNA is an example of a polynucleotide that is composed of the monomers, which are called nucleotides. So nucleotides are actually composed of 3 parts. So there's 3 parts to a nucleotide, and that's pictured here. The 3 parts are phosphate, a 5-carbon sugar. This is deoxyribose here in purple. That's deoxyribose. And then the third component is called the base. Sometimes this base is referred to as nitrogenous, a nitrogenous base. That just means that there's nitrogen in that chemical molecule. You see all those nitrogens there? So that would be the third component. You see they're all covalently bound together. Also take note of something else. The phosphate, you recall phosphate, and we've seen something similar to this. If you recall when we studied the structure of ATP, adenosine triphosphate. So adenosine triphosphate has a very similar structure here. And the phosphates have a negative charge. Notice that? So on this oxygen atom, we get a negative charge. And that's going to be an important thing to realize when we discuss biotechnology and some properties of DNA. So DNA has a negative charge as a result of the phosphate group in the nucleotide. So the bases, the nitrogenous bases of DNA, they are variable, and there's 4 different nitrogenous bases. Sometimes we refer to this as the alphabet of DNA, where there are 4 letters that make up the alphabet of DNA. You may have heard them before. So A, which by the way stands for adenine. So you should know the full name of the nitrogenous base as well as the shorthand letter that we use to designate adenine with a capital A. Guanine, guanine is designated with a capital G, cytosine with a capital C, and thymine with a capital T. Notice in this chart, they've grouped the 4 nitrogenous bases into 2 categories. So be aware of this term, purines. So purines refer to guanine and adenine. And notice that the purines are composed of double rings. So there's double rings that make up the purine nitrogenous bases, compared to cytosine and thymine are both examples of what are called pyrimidines. And the pyrimidines have 1 ring. So they have a single ring. And this is important to realize for the pairing, the base pairing that exists when the DNA molecule forms a polynucleotide and we get the 2 strands, the double strands, connecting with each other. When they connect with each other, we never pair 2 large double-ringed nitrogenous bases together because those would not be able to correctly hydrogen bond. In the same way, we don't put the 2 small pyrimidines together. Instead, in the base pairing, there's always a specific arrangement to how that works. And the way it works is that thymine, or the T, always base pairs with adenine. So there's a base pair interaction between adenine and thymine, and there's a base pair interaction between guanine and cytosine. In this figure, we take a look at the double-stranded DNA molecule. So we just learned that a nucleotide is composed of 3 parts: deoxyribose, the sugar, let's put an S there for sugar, and phosphate. So here's the phosphate, a P for phosphate. And then there's some nitrogenous base. So here they're telling us this is thymine, so we'll put a T there for thymine. So that means, for example, this would be 1 nucleotide. At the beginning of the semester, we learned that when we take a monomer, like a nucleotide, the analogy for a monomer is it's like a Lego. It's like a Lego brick. So you have this Lego brick and you connect it with another Lego brick. So here's the other nucleotide. And we end up with a polynucleotide. We see a connection here, okay, between these 2 nucleotides. And then we see on the opposite side, we see the complementary base. So remember, adenine and thymine are always hydrogen bond. So notice where those dotted lines are connecting the two strands of the DNA molecule. So those are referred to as hydrogen bonds, if you recall. So let's erase these marks and take a look at some other things I'd like you to notice from the DNA picture. So notice that there's this symbol 5' on this side of the DNA molecule, and then on the opposite side of the DNA molecule, on the other strand, we have a 3'. So this is a terminology related to the orientation of the DNA molecule. So the 5' refers to the 5th carbon that's attached to phosphate. So the way we do that is that if you look at the sugar molecule, there's carbons. In organic chemistry, these little nodes represent carbons. We talked about that when we talked about glucose in cell respiration, if you recall. So this is the first carbon, we have second, third carbon, fourth carbon, and then here's the 5th carbon. And that 5th carbon is attached to a phosphate group. So we call that end, the end where the phosphate is sticking up, we call it the 5' end. And then notice in the next nucleotide, we have the 3' end. So 3' end means that's the end where we just have the 3rd carbon pointed down. This 3rd carbon is attached to this oxygen-hydrogen group. We call it a hydroxyl group. So if the 3' hydroxyl group is pointed down, we call it the 3' end. If the phosphate attached to the 5th carbon is pointed up, we call it the 5' end. Notice 5' end, if we go to the complementary strand, we're going to have the 3' end. And then we'll have the 5' end that's opposite the 3' end. We call this orientation of DNA, we call it anti-parallel. What that means is if you understand the concept of parallel, so parallel means that if we said we have two parallel lines, which basically DNA is, it looks like a ladder, okay, a twisted ladder making up the double helix, but a lot of times we refer to it as a ladder where the ladder part represents the sugar phosphate backbone of DNA. And then we have the rungs of the ladder representing those hydrogen bonding between the nitrogenous bases. So we have the two strands of DNA that they are parallel. They're running, side by side. However, they're running in opposite directions. So if we would label the top of this DNA ladder 5', this side of the DNA ladder would be 3', and then the opposite strand would have the opposite orientation. So the concept of anti-parallel is an important concept to understand in the structure of DNA. So what that means is that the two strands of the DNA molecule are oriented in opposite directions. Which is why we show the orientation 5' to 3' on one strand, and the opposite strand, the anti-parallel strand, runs 5' to 3'. And this is also important to understand in the copying, the replication of the DNA molecule. So we're going to look at that next. So in the replication of the DNA molecule, this is an enzyme reaction. Can you think about when the DNA molecule would be replicated within cells? When would this be happening? That's right. This would happen in the S phase, synthesis phase of interphase. So in the last unit, we talked about how the DNA has to be copied. So the DNA is copied, sister chromatids form, and the DNA is ready to be split through a process known as mitosis. So this is a process that's constantly happening in our cells as new cells are formed as a result of mitosis. So the DNA has to be easily copied. Now, having the hydrogen bonds connecting the two strands is an important chemical bond to notice because remember, hydrogen bonds are very weak. So it's easy to pull apart the hydrogen bonds of the DNA molecule. So what happens in the replication of DNA? Well, here's how we would write out a strand of DNA. So we would write out that we have a 5' end, and then we would actually just write out the sequence of the DNA nucleotides. So let's say we have a sequence like this. So this would be one strand. So one strand would have a sequence. We would call this a sequence of DNA nucleotides. And we're labeling one end 5' and the other end 3' because that's how the orientation of the DNA molecule would look. And then the complementary strand, we would say there's a complementary strand, would have the opposite orientation. So you'd always write 3' and then we would, over here, we would write 5'. So they're opposite orientation, remember, anti-parallel. Now, let's write in what the complementary sequence would be based on the DNA base pair rules. So the base pair rules are, again, adenine pairs with thymine, cytosine pairs with guanine. So if we have an A, we would write a T in the complementary strand. A T, we would write an A. A C, we would write a G, and so on. Let's go ahead and fill that in. So this would be a simplified way to represent the DNA molecule, where we're just looking at the sequence of the nucleotides. So now we're going to replicate the DNA molecule. So we're going to replicate it. This involves an enzyme. So recall, enzymes are super important in biology. These little molecular machines that copy the DNA code and do many other things as well. But this one is the one that copies the DNA code, and it's called DNA polymerase. So that's a name you should know. Remember, enzymes almost always end in ase. And so that's a clue. We're talking about an enzyme here. So this is the enzyme that copies DNA. In cells. As far as we know, every cell on Earth, because all cells on Earth have a DNA genome, all cells have DNA polymerase. So bacteria do, plants do, animals do. It's a really universal and very important enzyme for the job that it's going to do here, which is to copy the DNA molecule. So what happens is there's actually a pretty complicated multi-step situation with DNA replication. Actually, the truth is there's a collection of at least 5 different enzymes that facilitate this process. For the purpose of our class, though, let's not worry about all of the details, okay? So the only enzyme that I want you to know for the exam would be DNA polymerase. But I do want you to understand the steps of what happens in DNA replication. So what happens is the DNA molecule, if you can think of it like a zipper, so if you think of it like a zipper, and then you unzip, think about like a coat or a jacket that you have with a zipper on it. And when you have that zipper zipped up, then the teeth are connected, right? The teeth of the zipper are connected. But if you unzip, your jacket, you unzip your zipper, now you have teeth on both ends, okay? Or exposed areas of your zipper on both ends. So that is actually what happens. So this DNA molecule unzips. So we'll go ahead and rewrite the DNA molecule. Let's leave some space because we're showing that we have this closed up zipper at the top and we're unzipping our zipper. So let's give ourselves some space. So the complementary strands, remember there's hydrogen bonds holding the strands together, and those hydrogen bonds, because they're weak bonds, are going to break and allow the zipper to unzip. So DNA polymerase actually can only add new nucleotides from the from a 5 to 3' direction. It's actually picky about it. So in studying DNA replication, there's actually some complicated steps that we're really skipping over here for the purpose of our class, okay? But we'll go ahead and say here that it's this 3' end DNA polymerase recognizes, and then it actually is going to build in that direction on the top strand. So on the top strand, it'll read, we would say that's moving in a 5 to 3' direction. So it basically recognizes the 3' end and then starts a new 5' end and then starts to add the corresponding complementary nucleotides that would match here. It'd be complementary by the base pair rules. Then we would have a 3' here. And then the bottom strand would also be copied by DNA polymerase, but it would be this 3' end that's recognized, and a new 5' end would be built, and we would be building from 5 to 3' direction in the new strand. So we would have an A and then a T, and so forth. Then we would end with a 3' end. So now we have two new DNA molecules that were built from this original DNA molecule. If you went with two different colors, that helps because we see the original DNA molecule had everything written in black, and then we zipped apart the two black strands. Then DNA polymerase comes in and building in opposite, building 5 to 3', so moving in this case would be from right to left in this picture. The top strand, the new strand would be in red. And then moving in the 5 to 3' direction at the bottom strand, we also see a new strand here in red. So we end up with two new DNA molecules. Let's draw that one other way that I think might be helpful. So again, let's think about DNA like a zipper, or you can think of it like a ladder where the rungs of the ladder represent the base pairing between the nitrogenous bases of DNA. So this would be a single copy of DNA. This would be a single DNA molecule. So DNA is going to first unzip. Remember, it's like a zipper. So if you have your zipper coat, you unzip it, you expose the sort of the teeth of your zipper. What is that analogous to here? Well, it's analogous to the nitrogenous bases. And then the new DNA is going to be synthesized by an enzyme, because remember, chemical reactions inside cells, they don't just happen magically. The DNA is not going to pull itself apart. It's going to stay intact unless it gets an interaction with an enzyme. And remember, again, it is a collection of enzymes. I'm oversimplifying it, but this main enzyme is DNA polymerase, and DNA polymerase comes along and builds a complementary sequence. So using the base pair builds the complementary sequence, and it does that for the top strand, and it does that for the bottom strand. So what happens is now we have two DNA molecules, and they are sort of hybrids. You see that? So the new DNA is in red, and the old DNA, or another word for that is template DNA, is in black. We have a term for that you should be aware of. The way we describe the replication of DNA in cells, we describe it as semi-conservative. So we would describe the replication of DNA using this terminology, semi-conservative. What does that mean? Well, the word semi means half, and conserve means to keep. So it means that when the DNA molecule is replicated, half of that molecule is kept or conserved and used as template. Remember, that's the black strand in both pictures. So the black strand in the two molecules that are being built is conserved or kept the same and used as template so the DNA polymerase knows what nucleotides to add to the complementary strand. So let's see if we can write that out. So semi-conservative means half of the DNA molecule is used as template in synthesizing, which is just a fancy word for making, for synthesizing complementary DNA. So semi-conservative, half of that original molecule is conserved in the copying of the DNA. And that's it for this video. Hope that was helpful.